Okay, everyone. So I hope that I am live and I am clearly audible and visible to each and every one of you. Yes or no? So before starting the session, let me check that am I live or not? Am I clearly audible or not? Right? So. Yes. Great. Let's quickly start this session, people. So today what we are going to have is that we are going to, this session is all about the genetics, right? So at 4 o'clock what we are going to do is that we are going to do genetics as we were doing, and 6:30 we are going to do the Z scheme today, which unfortunately we left in a half in yesterday's class. Okay, I hope that all the classes, today's class will go very smooth. Clear? Okay, mark your attendance, people, everyone. If you are present in my live class, mark your attendance, say present, absent. Clear? Okay, great. So let's quickly start with this session. Today in this particular session, we will be discussing about the post Mendelian genetics. Okay, we discussed about the Mendelian genetics. We discussed about dihybrid cross, monohybrid cross, and their laws, that is law of dominance, law of inheritance, and everything. Now it's time to leave Mendel and start the post Mendelian genetics. Remember that this post Mendelian genetics is basically what? These are the time period, or these are the things which were not studied by the Mendel. Okay, these things were done by someone else. So post Mendelian genetics. This is also a very important topic from which the question comes, and it's very important to understand. In the post Mendelian genetics, the very first thing we need to understand is the intra-gene interactions. Okay, the first thing which I'm going to explain you will be intra-gene interactions. Now, I hope that you are able to distinguish between what is gene and what is allele. Clear to everyone? Allele is what? Allele is a different form of gene. Okay? Allele represents what? Allele represents the traits, while gene represents the characters. If you have any kind of confusion in this, I'll explain you again. So see, if I'm talking about the plant height, right? Suppose I talked about the plant height. So plant height is what? Is it a character or is it a trait? So my answer will be the plant height is what? It is a character. Yes, sir. Let's represent it with C. Plant height is character. Now, if I am going to talk about what kind of plant height we have. So, we have two types of plant height. One is tall and the other one is what? The other one is short. Clear? So, this is what? This is the trait, right? Tall and short is what? It is the trait. Similarly, we have what? We have gene for plant height. Okay, we have a gene for plant height. Now this gene contains different forms, like a gene for the plant height contains an allele for tall character and a allele for short character. That means types of genes or forms of genes are the alleles. I hope that this thing is clear to everyone. What is the basic difference between the gene and the allele? If it's not till clear, go to the previous classes, watch that recording, because in that recording we have discussed these terminologies very clearly. Clear? Now, let's get back here. So, we were discussing about the intra-gene interaction. Okay, sir. Now, intra-gene interaction means what? Intra-gene means interactions within the gene. Yes or no? Inter-house competition is what? In your school, inter-house competition takes place, and inter-house means what? Competition between two houses. If I am saying that intra-house competition, that means let's suppose that you are from a house named Coral. Okay, I was in the Coral house during my school days, and this house was like, I don't remember when our house won something or something not. I was not the head, don't worry. I was just a member. of this particular thing. So my house never won, unfortunately. There were 4 to 5 houses, coral, topaz, emerald, and there were one more, sapphire. So I was in coral and coral never won. So let's get back here. If suppose I am in coral house and inside the coral house, we are having a competition that who is going to lead this coral house. So that is what? Competition inside the coral house, which means intra-house competition. Inter means between two houses, intra means inside the house. Clear to everyone? Okay. So if I'm saying that intra-gene interaction, that means something inside the gene. And inside the gene is what? Basically the alleles. So can I write here? Or this can be called as inter-allelic interactions. Okay. Or this can be called as inter-allelic interactions. Inter-allelic interactions. One second, people. This can be called as inter-allelic interactions. The first case we are going to see this. Clear? Okay, sir. Now, the very first example of this is what? It is incomplete dominance. What's the meaning of this heading, sir? Intra-gene interactions or inter-allelic interaction. This means that what is happening? Two alleles are interacting with each other, right? Two alleles is interacting with each other. This is the intra-gene interactions or the inter-allelic interaction. So, the very first thing which we are going to read here will be what? Will be case of incomplete dominance. Will be the case of incomplete dominance. Now, what is this incomplete dominance? Who is going to tell me? What do you know about the incomplete dominance? So, if I talk about the incomplete dominance, let's have an example. If I talk about the dominance, bio syllabus Vinika we will complete in January and we will complete the biology syllabus, and you had one more doubt about the about the zoology and botany, right? So don't worry, I will be taking zoology and botany both, okay? Don't worry, I'll take the responsibility to complete the syllabus, okay? Don't worry, no issues. Clear, Vinika? Great, happy now? Okay, let's start then. So now I am talking about the incomplete dominance. Before moving towards the incomplete dominance, let's remember Mendel uncle who did some great experiment. So if I talk about the Mendel thing, so we had this one, yes or no? Capital T, small t, small t. And this made capital T and small t. Yes, this was what? This capital T and small t was a phenotype which is producing a tall plant. Yes or no? Yes, sir. So here what was dominant? Capital T was dominant. Yes. In case of any doubts, Vinika, what you need to do is that you just need to write in the comment section. I'll keep your doubts, okay? So capital T is what? It is a dominant allele. Yes, it is a dominant allele, and that is why it is showing its phenotype which is tall. Yes, sir. This is very much clear. We know this in the Mendel thing. But now, in case of incomplete dominance, the thing is something different. There is a lot of difference in case of Mendelian genetics and in this thing. So, for example, I am taking a plant, let's suppose a 4 o'clock plant. Okay, for example, let us take a plant which is 4 o'clock plant. Okay, yes, sir. Now, this 4 o'clock, how many of you know the scientific name of this 4 o'clock plant? Anyone? Anyone? Anyone in the comment section? Fast. What's the scientific name for this particular plant? It is Mirabilis jalapa. Okay, it is Mirabilis. I think so. This is the spelling only. Mirabilis jalapa. Okay. Now, this plant comes into two colors. What colors, sir? It comes into red and white color. Okay, it comes into red color and white color. So, we have red which is dominant and white as recessive. Okay. Red is dominant, and remember here also we have taken pure homozygous plant. Homozygous was what? Homozygous was those conditions in which both the alleles were Same. So in here also, if I'm saying about the homozygous condition, so we will have capital R and capital R here. In the similar manner, again, they have also taken the homozygous recessive plant also. This will be small r and small r. Okay, sir, great. Now let's make the gametes. Guys, if you have any doubts in these terms, no, small terms like homozygous, heterozygous, or let's suppose how to form gametes and anything, I have taken a lecture on this thing. I'll put the link in the comment section after this class. You go and revise those things, okay? The concepts will be very much clear, okay? And in case you are having any doubts right now, comment section is there, right? Okay, great. Now, just give me a thumbs up if you are understanding everything. Now, this is what? This is capital R. Where is my other hand? Yeah, see, this is the capital R, okay? And this one is the small r. So, gametes will be formed. This is going to give what kind of gametes, sir? Only one type of gamete we are going to get, which is capital R. Here, one type of gamete, which is small r. And then they will fuse and form capital R and small r. Right now, hi Shri Shai, how are you? Right now, you will be like, sir, did you did that joke? Did you crack that joke? Same thing you have done like Mendel uncle did. Yes, the Mendel also did the same thing. So, what's the difference? But the difference is, if I ask you, what will be the phenotype? Vinik and Shri Sai, what will be the phenotype? So, you will say, sir, the phenotype will be red in color. And then I'll say that no. Here the phenotype is not red in color, rather the phenotype is pink in color. The phenotype is what? Okay, great Sri Shai. 4 to 5, class 12, 6:30 to 7:30, class 11th. Okay, everyone? Okay? Chalo. So, capital R and small r is giving here the pink phenotype. This was supposed to give the red phenotype, but it gave pink phenotype. So, this is the case of incomplete dominance, in which what? This dominant gene, this dominant allele is what? This dominant allele is little bit suppressed in presence of this particular recessive gene. This is not a normal case. This is an exceptional case. So, in incomplete dominance, you have to remember this example of Mirabilis jalapa. Clear to everyone? Okay. Now, you might be thinking, sir, that in every cases, are we going to get pink color? Is it like that when we mix red and white color, we get pink? So, all the now generations will be pink, 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 pink. It can happen, right? If suppose that this mother and this father reproduced and both their colors got mixed, and due to mixing, the progeny is what? The progeny is having pink color. So, when that pink progeny will reproduce, all its babies will be having pink color. Yes, this is true. But this didn't happen. When we did selfing, what is selfing, guys? Sir, will you complete syllabus detail? Can we get out of you? Yes, definitely. Many of my students are in medical college. You just have to trust me, okay? It will take little time for us to build a connection, but don't worry. Everything I'll make your biology super duper easy. Okay? You will feel love with biology. Clear? No issues, Shri Shai. Koi baat nahi, next bar sir on time, okay? Chalo. Great. Now what? Now sir, let's do the selfing. What's the meaning of selfing? It's just a humble request to you guys that keep interacting with me, okay? If you are not understanding that thing, see, I'm here to teach you. If you are not able to understand anything, let me know. If you are able to understand a concept, let me know, okay? Whatever you need, let me know, okay? Done. Great. Now, let's move on. So, we are going to do selfing now. What is selfing, sir? Selfing is the self-pollination. We took this particular child, not child, I should say progeny, this particular progeny, and his or her siblings we have taken, and we did the self-pollination. So, we will be having capital R and capital R and small r. So, at this point of time, why we are doing the selfing? We are doing the selfing to just have a visualization that is this pink color mixed or not. So when we have done the selfing, we got what? We got this kind of genotype: capital R, capital R, capital R, and small r and small r. Remember that F1, F2 generation in Mendelian cross, same way we are going to do, okay? Should I make you understand the Punnett square again? No, if you haven't done it, go and watch the first lecture, okay? Clear? Great. So now we will figure it out. Like capital R, we saw red thing. Again, small r, we saw white phenotype. And again, capital R, small r gave us the pink phenotype. That means what, guys? That means the color is not mixed. The color is not mixed. Pink color is only and only coming when there is the heterozygous condition of capital R and small r. That means what's happening here? What's happening here? It's basically in presence of small r, in presence of recessive allele, capital R's strength is kind of decreased. Capital R is not able to show its full color, right? So this is what incomplete dominance. Say, any doubts anywhere? Shrishai, Vinika, any doubts anywhere? Let me know. Is it clear? Selfing is clear? Okay. So can I write here? This is not the case of blending. Incomplete dominance is not the case of blending. Where should I write? I'll write here, okay? It is not the case of blending. It's not blending. Why? Agar blending hota, just imagine if you have blended the colors, how you would have again got red and white color separately, right? So the color is not blending. The reason behind the pink color is due to the condition known as incomplete dominance. Clear to everyone? Great. Yes? Now, it is not the case of blending. It's not the case of blending. Okay? Should I move forward? Let me know fast. Let me know. Now, another example for this thing is what? Another example which can be asked in your exam is snapdragon flower. Okay? It is what? It is snapdragon flower. Snapdragon flower. Okay? So, how can the question be put up? Mirabilis jalapa is an example of incomplete dominance. Similarly, the snapdragon flower is an example of incomplete dominance. So, whenever the question is being asked with these type of organisms, in your mind, you should have this thing that, okay, this is the case of incomplete dominance and I'll be solving the question keeping this in mind that they are the case of incomplete dominance. Okay? In snapdragon flower, what's the scientific name? The scientific name is Anthridium majus. Okay, Vinika, for sure. I'll upload it. Okay, I'll upload it. Don't worry. Anthridium. Oh, what I'm saying? Anthridium majus. Okay, it is the scientific name. Do remember the scientific name, okay? Because in exam, they're not going to give you the direct snapdragon or 4 o'clock plant. They are going to give you the scientific name, okay? Again, another example for the incomplete dominance. Okay, how many of you have seen cow? Obviously, sir, we all have seen cow. What you are asking, sir? What is this behavior? Huh? So, see, example, body color of cows or fowls, okay? So we have seen some cows are having black and white spot, okay? How, sir? Because of the incomplete dominance. Yes or no? How? See, let's suppose the parents were black, capital B. The parents were black, okay? And then we have a white cow, which was small b, which was white, right? Okay. Okay, sir. Now make the gamete. What will be the gametes be formed? They are going to form the gametes as capital B from this black and small b from this white. Okay, so we are going to get small capital B, small b. Yes or no? And this color is going to what? This will be this will be having the patches of colors like black and white patches. Clear to everyone? This will be having the patches of color like black and white. Okay, done. This can be also the one example. Okay, everyone? Clear? Great. Now, let's move on and let's talk about the another examples or the another case. Incomplete dominance, everything is clear or not? Give me a thumbs up if it's clear. How many examples are? 4 o'clock plant, Snapdragon, 4 o'clock plant is known as Mirabilis jalapa, Snapdragon is known as Anthridium majus, and we have the body color of the fowl. Clear to everyone? Yes or no? Shri Sai, Vinika, clear? Great. Now, let's move on with the another example, which is what? Which is codominance. Which is codominance. Codominance. Clear? And in this codominance is what? It's like, you know, let's suppose that you and your best friend, right? This is you and this is your best friend. Okay? This is you and this is your best friend. Every time when we go to school, like we used to do these kind of things. I don't know about you all guys, but we were like, bro, when we will grow up, we will make a company, half of the percentage you take and 50% I'll take. So what we have done? Codominance, right? I have given 50% stake to my imaginary friend and I have taken 50% of mine. So here also, what they are doing is that the alleles are like, okay, you take 50%, I will take 50%. Clear? So this is what this is the codominance case. There is no mixing. Remember, there is no mixing like in complete dominance. Rather, both the alleles are having what? Both the alleles are having equal percentage. In this case, the example is codominance. I will going to explain, you will be able to understand this thing in a much better manner. Okay. So in case of codominance, let's suppose we are going to change things. How we are going to change things? See here. If I am writing B1, so B1, let's suppose friend 1 and this is B2, this is friend 2. Okay. This is what? This is B1 and this is B2. So in case of codominance, we are not writing B and small b. We have changed the pattern. This is called as B1, this is called as B2. Both with capital letters and writing 1 and 2. Clear? Okay, sir. Now what, sir? Let's suppose I want to write a homozygous condition in case of codominance. So let me write homozygous condition here. So how I am going to write? I am going to write it as like B1, B1. Okay. Or how I can write? I can write as B2, B2. Clear? Okay. Clear? This is how we can write the B1, B1 and B2, B2 as homozygous. How can we write heterozygous conditions, sir? See, in the heterozygous condition, we can write B1, B2, B1, B2. Okay. Like instead we were writing what? We were writing earlier, we were writing B and B for the dominant one, small b for recessive one, right? And capital B, small b for the heterozygous condition. But in case of codominance, we are not going to represent it like this. Rather, for everything, we are using the capital letters and we are just numbering it 1 and 2. So B1, B1, B2, B2 and B1 and B2. Clear? Okay. Great. Now, if suppose B2 is recessive, then also we are writing it as capital B only, just with the subscript of 2. Clear, Vinika? Clear to everyone? Okay. Great. Now, let's take an example. Let's take an example of what? Of bull. Okay. Of bull. Clear? Let's take the example of bull. We have blacks. Oh, sorry. What I did is that this example will be here. I'm so sorry. I explained this example here. 1 second, people. 1 second. This example we will be doing here. This is the example of codominance. I am very sorry. See. So we are taking again that example which of a bull and a cow. Okay. So we have bull. Bull is suppose black in color. Bull is suppose black in color. So I am going to write it as B1 and B1. Bull is suppose black in color. So I am going to write B1 and B1. And here we have cow which is white in color. Cow which is white in color. Okay. We have bull as B1 and B1, and we have cow as white color which is represented as B2 and B2. Clear? Great, sir. Now, can you make the gametes here? Can you make the gametes here? Yes or no? Yes, you can make. From here, you're going to get B1 as your gamete. You're going to get B1 as the gamete. And here, what you're going to get? B2 as the gamete. Okay. When these gametes fertilizes, you are going to get what? You are going to get B1, B2. You are going to get B1 and B2. The phenotype which you are going to get is what? It is the round color. It is what? It is the round color. What is round color, sir? We don't know. Round color is basically what? It is the patches of black and white color. Round color is what? Patches of black and white color. Okay. Patches of black and white color. Clear? That's great. That's great, Shri Shine. Vanika, are you there? So round color, patches of black and white color. Okay, great, sir. Now, see, in animals and in human beings, we can't do self-pollination or selfing, right? It's unethical also, and practically also it's not possible. Let's suppose there is a person who is experimenting on human beings. So he has to wait for 9 months, first of all. Again, that 9 months for the baby will grow. Again, some 12 to 13 years till the baby reaches puberty, right? So, in humans or animals, like doing selfing is kind of very different. But here we are just considering that, let's suppose we have done the selfing. Let's suppose we have done the selfing. So, selfing means what? Selfing means cross between the F1 generation, yes or no? Cross between the F1 generation. So, we have here B1, B2 reproduced by B1 and B2. Clear? Okay. Okay, sir. Now, how many types of gametes will be formed here? How many types of gametes will be formed? See, this one is going to give you one gamete, which is B1, and the other gamete which you are going to get from this particular will be B2. Okay. Similarly, here also you are going to get B1 and B2. Now, to make the gametes, the best thing to avoid mistake is to make the Punnett square. The best thing to avoid mistake is to make the Punnett square. So, let us make the Punnett square here. How we can make the Punnett square? See, a very simple idea. How many gametes you can see? 1, 2, 3, 4, right? 4 gametes is visible. 1, 2, 3 and 4. So, what you do is that tuck and tuck. Clear? Make the 4 boxes. Now, arrange these gametes here. B1 and B2. Okay, for one parent, let's suppose this is male. So, for male we have arranged, and this is female. So, let's suppose we have arranged for female here as B1 and B2. Now, let's make the gametes. Sorry, progeny. So, B1, B1. Okay, sir. B1, B1 we got. We got B1, B2. Okay, sir. B1, B2. Here also B1, B2. Yes. And here we got B2 and B2. Understood? So, how many things we have got here? How many total progeny we have got? 1, 2, 3, 4. Among this 4 progeny, we have what? We have B1, B1, 1 and 2 B1, B2 and 1 B2, B2, right? Okay, sir, great. So, this B1, B1 is homozygous. B1, B1 is homozygous. So, if it was homozygous, it will give us the black color. Right? This B2, B2 is again homozygous, but B2 was representing the white color. So, we are going to get white color. But here what? Here we are going to get the patches of white and black color. Patches of white and black color. Black color. Okay, clear to everyone? Vinika and Shrishai, is it clear? Any doubts anywhere? Let me know. Okay, patches of black and white color. Clear to everyone? Great. So this was one example of codominance. Now, the next example which we are going to do is very, very important. That is what? That is the example of human blood group system. That's the example of human blood group system. Okay, great. Now see, this one is very important, you know, and very interesting also. Human blood group system. Can Ron be red and white? No, like combination of black and white, we say it as Ron color. Okay, combination of black and white, we have given it a name, a pattern like Ron pattern. Like we do this na, so zigzag pattern. So just Ron pattern has been given to the name of that black and white. Okay, clear? Human blood group system. Now see, how many types of blood groups do we have? So sir, as of now we have A, B and O, right? Or we can add here is that AB. Yes or no? We have either A. Let's forget about positive, negative. Okay, let's just focus on A, B, O and AB. Clear? Okay sir, that's great. Now see, what's happening in the blood group, human blood group system is what? See, in human blood group system, there are 3 alleles which are controlling our blood, right? There are 3 alleles which are controlling our blood system. Okay, yes sir. So A, which alleles are controlling our blood group? IA, IB and IO, right? Okay, so in this case sir, if we are having A blood group, see, this thing we are going to read in much detail in multiple alleles example, but a very short thing I'm going to tell you, like why we are keeping this in the codominance also. See, so A we have IA, okay, B we have IB, okay, O we have IO, and AB we have IA and IB. So can you see here? Can you see here? This is what? This is the case of codominance. This is the case of codominance. Okay. Blood transfusion and everything, how IA and IA is formed, how IB and IB is formed, we are going to discuss in the multiple allelism. But as of now, this is what? This is an example of codominance. Here, character of A allele is also being shown, character of B allele is also being shown, just like here, right? Here in Ron, what was happening? Character of B1 was also shown, character of B2 was also shown. So in the similar manner, if a person is having a blood group AB, we can call that case is of codominance. Okay, clear to everyone? Great. Now, one more thing, I am going to give you an example of heterozygous condition for sickle cell anemia. Okay. Another example is sickle cell anemia. This is just the short example. We are going to learn this thing in very detail in multiple allelism. The main example of blood group, if I am going to ask you, the blood group is the main example for what? Multiple allelism. But IA, IB can be the example of codominance. Okay? Clear? Shrishai and Vinika? Great. Now, sickle cell anemia is what? Sickle cell anemia is also an example of this codominance. How, sir? See. First of all, I am going to give you some detail. Let's suppose HbA. If a person is having this allele, that means it is normal. Normal hemoglobin, normal RBC. HbA means what? Normal hemoglobin and normal RBC. No issues. No issues with this person. Okay. Great, sir. Now what? If a person is having HBS, today I have a very great surprise for you at 6:30. I have made a story for you all so that you can easily understand that photo system or Z scheme. Okay, and I hope that yesterday, like yesterday's class, we don't have any problem at that time. Okay, so HBS is what? HBS is this allele which is causing the problem. HBS is the allele which causes the problem. So it is what? In this, sickle cell mutation has happened. HBS is sickle cell mutation has happened. Mutation has happened here. Okay. And hemoglobin will be what? Abnormal. Abnormal hemoglobin. Again, I am saying you, everything we are going to discuss this sickle cell anemia also in detail. Just we are going through an overview right now. Don't worry. If you are thinking like, sir, you didn't talk about the mutations, how, where, when, everything we are going to go in very detail. Right now, whatever which is needed, I am telling you. So here the this hemoglobin will be what? It will be abnormal, and RBC shape will be what? Sickle shape. RBC shape will be sickle shaped. What is sickle shaped? Something like this, okay? Sickle shaped. And what's the problem, sir, if the shape of the RBC will be sickled? So number one thing, it is not going to have lots and lots of hemoglobin, so oxygen will be less. And the sickle cell anemia is what? It gets stuck into the blood vessels, which can cause blood clotting and many diseases. Okay, so sickle cell anemia is a very serious problem. So what happened is that, let's suppose a person is having this condition, HbA. So it will be normal, no issues, right? Because HbA is what? HbA is a normal gene. HbA is a normal gene, so it will have no issues. Okay, sir. Normal gene. If a person is having HbS and HbS, sir, so this person will die. RIP. This person will not survive. RIP. Okay. But sir, if a person is having HbA and HbS condition, so what? So 50% of the RBC will be normal, 50% RBC normal, and 50% RBC will be affected. 50% RBC will be normal and 50% RBC will be affected. Clear? Okay. Affected. 50% RBC normal and 50% RBC affected. Clear to everyone? So how many examples we have seen for this particular thing, which is the codominance. One is the human blood group system. In human blood group system, if I ask you that IA is the example of codominance, no, it's not the example of codominance. This IB blood group, IA and IB is the example of codominance. Okay. Then we saw the sickle cell anemia. In sickle cell anemia, we saw there are two alleles, HbA and HbS. HbA normal, everything will be very fine. The boy will be chill, pill, dancing everywhere, no issues. But if there will be HbS and HbS, it will be that will happen just after the birth, or the baby will be born, like it will be born dead. Okay. So in this case, there is no chance of survival. But in this case, which is HbA and HbS, yes, exactly, carrier. HbA and HbS, what is going to happen? 50% will be normal and 50% will be affected. Clear? Okay. Carrier and everything we are going to read in very detail. I am very happy that you said this, Shri Shai carrier. Okay. Clear? Great, sir. Now, should we do some questions? Should we do some questions? Okay, I'll plan a session for the questions. Let's complete multiple allelism today. Okay, and tomorrow at 11 o'clock, will that be fine? It's Sunday, but okay, I'll not take off. It's fine for me. Tomorrow at 11 o'clock, we'll solve the questions from Mendelian genetics, which is monohybrid dihybrid cross. And then we will solve the questions from codominance and incomplete dominance, and then we'll solve from the questions from multiple allelism. Will that be fine? All the questions at one class, and we will clear out all the concepts. Okay, so now let's do the multiple allelism. But if you're coming to tomorrow's class, I request you to revise all the things, so that if you're going to have any doubts, that will be cleared. Okay, otherwise I will be like, I have gave the question, I'll ask you the answer, and you will be like, sir, we have not revised, you can do it. No, so that should not be done. Okay, so you revise and come, so that I can give you the question and you can give me the answer. In case you are going to have doubt, I'll be here, but yeah, the revision is necessary. Okay, now let's go with the multiple allelism. As the name suggests, okay, leave biology. I'm going to give you one example. Forget multiple allelism right now. If I say that, what happened to my screen? Okay, 1 second. If I say that, let's study multiple colors. Multiple colors. So what will be your answer? What will be the answer? Yeah, sir, we will. This means that we are going to study about some red color, blue color, green color, black color, right? Multiple colors. If I am saying multiple allelism, that means what? We are going to study about the multiple alleles, right? See, biology is very easy. We make biology hard by thinking that, oh, multiple allelism, I don't know, sir, what is going to happen here? Multiple allelism, as the name suggests, many alleles. Okay, if suppose that one gene represented by more than 2 alleles. Here, if I am talking about plant height, okay, if I am talking about plant height, so how much variations do we have? Sir, one is tall, the other one is short. Yes or no? How many variations? 2 variations. Is this the case of multiple allelism? No. If suppose I am talking about the human blood group, if I am talking about the human blood group, so what is this human blood group? So sir, human blood group, we have seen that there is some more alleles, like IA is giving the A blood group, IB is giving the B blood group, sir, and IO is giving the O blood group. Okay, IO can be written as like this also. Okay, and this we can see that there are total 3 alleles here, like 1, 2 and 3. This can be the case of multiple allelism, right? A single character is having more than 2 traits, or a single gene having more than 3 alleles. Clear? Clear to everyone? That's great, Srishai. Revise and come. Okay, clear? So this is the case of multiple allelism. Let's write some of the theoretical points here. Should I make you write? Srishai, you are in class 12th or class 11th, or are you a dropper? See, so one gene, what's the case here? One gene represented by more than 2 alleles. More than 2 alleles. This is the case of multiple allelism. Okay, 12th, great. One gene represented by, so we have to crack NEET this year. Okay, no dropping. Okay, this year only we are going to crack NEET. One gene represented by more than 2 alleles. Great, sir. Now what? If suppose I ask you that how many alleles can be possible, right? So see, if I am going to talk about number of possible genotypes here, number of possible genotypes here. So let's suppose I have told you, like Srishai, we have 3 alleles. Okay, sir, you said, and then I asked you that how many types of combinations you can make from here. Like you will be like, sir, one combination IA, one combination IB, one combination II, and one combination something. But this is kind of very hectic, right? One by one if we will say, and one by one if we will able to write, we might make mistakes. So we have a formula for this. What formula? We have a formula which says n plus 1 upon 2. If you need to find out that how many types of genotypes is present in a case. So there is a formula n plus 1 by 2. Sir, what is n? N is the number of alleles of a gene. Number of alleles of a gene. So in this case, how many alleles are there? 3 alleles. Okay, so we will put 3. I am very weak in maths, but don't worry, I know these small calculations. Okay, so 3 plus 1 upon 2, what will be it? 3 plus 1 will be what? 4, 4 3s are 12, 12 by 2 will be 6, yes or no? So in total, we will be having 6 genotypes. In total, we will be having 6 combinations, yes or no? Yes or no? Say me. Yes, obviously we can have 6 combinations. I'll show you, don't worry. Right? 6 combinations in total we can have. Great, sir. Now, one more thing, people. One more thing. We all are diploid. Yes, sir. Is there anyone who is haploid here? No, no one can be haploid. Yes, 6 genotype. So we all are diploid. So if I ask you that what can be the maximum, listen to my question very carefully. What can be the maximum alleles I can have for a single character? How many alleles can I have for the blood group? Can I have 3 alleles in my body? IA, and I also. Can I have that? No. Because we are diploid and we can have only 2 sets of alleles. Yes or no? So, in case of this multiple allelism, although we are having 3 alleles in total, but a single person will have only and only 2 of them. Because 3 is not possible. Now, how can you accommodate another set of allele? We are not triploid, we are not 3n, we are 2n. So, for one character, we have 2 alleles. Clear to everyone? Any doubts in this? Is this concept clear? Say, is this clear? Great. Okay. Now let's move on. Okay, I'll write this as a statement so that you can write it in your notes. Where should I write? Here I am writing. Okay. So let's suppose a diploid organism can maximum have how many alleles? Have only 2 alleles of a gene, right? And suppose now you have to study this. You have to study IA, IB and I. So you can't do your study on a single person. You require the whole population, right? So to study multiple allelism, we require a whole population. Clear? Great. Okay, that's great. So now let's move on and let's make the genotypes of all the alleles, let's write the genotypes of all the blood groups which are present here. Okay, see. So if I am saying that IA will produce what, sir? IA will produce A blood group. Okay. IB will produce what? IB will produce B blood group. Okay. If I is there, it is representing O blood group. Okay. Now, a very simple example I am going to give you. A very, very simple and easy example. Just consider there are 2 terms. Okay. There are what? There are 2 terms. One is the antigen and the other one is the antigen. Okay. Let's not get back there. First of all, let us understand this thing. See. We have what? We have IAIA. First of all, let's write this, then I'll make you understand. Then we can have IAI. Then we can have IBIB. Then we can have IBI. Okay. See, my dog is also excited. And then we can have what? IA, IB. And then we can have small I. Okay. 6, right? 1, 2, 3, 4, 5, 6. See, that means the formula which I said was correct, right? Okay. Now, so we have this 6 type of genotypes here. Now, what happens is that there are two terms which is antigen and antibody. Okay, there are two terms which is antigen and antibody. Sir, we are very much confused about antigen, antibody, what is this and all. So, treat antigen as a nameplate. Treat antigen as a nameplate, okay? What is nameplate, sir? When you have your notebook in your school, when you submit the notebook, what do? How a teacher can recognize your notebook? By your nameplate. Yes or no? What I used to do is that I was a very naughty kid in my school. So whenever I don't do the homework or something like that, so I used to tear the nameplate of 4 to 5 copies, and then the teacher were not able to recognize that which person has done the homework or not. But you guys don't do these things, okay? So antigen is what? Antigen is a nameplate. Okay. Nameplate means what? Whatever it is written, suppose it's written Harsh. So you will understand that, okay, this is the notebook of Harsh. Suppose Sri Shai is written. So you will understand that the notebook belongs to Sri Shai. If suppose Ram Sham, anything is written, so the notebook belongs to that person. If suppose I write A here, if suppose I write antigen A, which means what? Which means what? It means A blood group. It means A blood group. Okay. So antigen, how you can recognize a blood group? By seeing its antigen. What is antigen, sir? Antigen is basically a set of protein which is present on the membrane surface of the RBC. RBC antigen ऐसे लगा के घूमता हर जगह, right? So antigen is just placed on the surface of the RBC, and when you are going to see the antigen, okay, it is A, so the blood group will be A. It is B, the blood group will be B. Clear to everyone? Antigen A. If suppose I said that there is antigen B, so then what you will going to say? Antigen B hai, so blood group will be B. If I am going to say no antigen, if I am going to say no antigen, that means what? That means there will be what? The blood group will be what? The blood group will be O. No antigen, that means blood group O. Clear to everyone? No antigen, blood group O. Great. Now what is antibody, sir? What is antibody? See, there is what? There is always one of kind of our friend which says that, see, you are my best friend, I hate him or I hate her, you don't have to talk with it. Okay. So A says what? A says I hate B. A says what? I hate B. So if antigen and antibodies are best friend. So A hates B. So antibody is also going to hate B. So it's right, anti-B. So in case of blood group A which is antigen A hates B. So the antibody which will be present in the blood group A will be what? It will be anti-B. Clear to everyone? It will be anti-B. Yes or no? Clear? Now, if suppose the blood group is what? The blood group is B. That means antigen is B. So his best friend antibody will hate what? Will hate anti-A. Okay. So it is going to have antibodies for A. See, here it has no name, right? It has no name. So it is going to hate both of them. It is going to hate both of them. Anti-A and anti-B. Zero, na? No name. No best friends. Nothing. So it will going to hate everyone. Okay. It is going to hate everyone. Clear? Understood? Antigen, antibody concept clear? Antigen, antibody concept clear? They were two best friends. If its name is A, so antibody can't be the friend of B, so it hates B. So antibody B. Blood group B, it hates A, antibodies will be of A. No name, no best friends, so it hate everyone. Okay, clear? Shri Sai, clear? Is there anyone in my class? Yes. Vinika, clear? Great. Okay, did you like this example? Did you like this example? Now see, this will be very much easy for us. Let me make this little small. Now my iPad is throwing some tantrums. See, what's the, one second. Tuk, 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 tuk. Yes, now I can select it. Okay. So now here we are going to write, in first we are going to write antigen. First we are going to write antigen. Then we are going to write antibody. Okay. Antibody. And then we are going to write what? Then we are going to write blood group. Then we are going to write blood group. Clear? Okay. Now, see how easy it will be. IA, blood group will be A. Okay. IA, blood group will be A. Antigen will be what? Of A. Antibody will be what? Antibody will be anti-B. Yes or no? See, now what? This was dominant, right? So, in this case, IA and IB is dominant and small I is recessive. Okay. So, this is recessive. These two are dominant. So, here we have IA and I. What we have? IA and I. This one is recessive. So, the blood group will be what? The blood group will be A only. Okay. And if the blood group will be A, sir, then we have anti-B and A. See? Now you won't be able to forget this thing, right? Again, we have B and B. So blood group will be what? Blood group will be B. Great. Blood group will be B. Antibodies will be what? Antibodies, the blood group B is going to produce antibodies of A. Clear? And antigen will be what? The nameplate will be what? It will be of B. Similarly, here also B, anti-A and B. Great. IA, IB. This is the case of codominance. This is the case of codominance. Oh, is that so? Yeah. We will also meet once. Okay. We will organize a meetup when we crack NEET. Okay. Now, AB is what? Codominance case. So now see, this case of AB, the antigen will be what? It will be A and B because it belongs to both of you. Like we do na sometimes out of curiosity. Okay, we are best friends. We are going to write a diary. One day you take the diary home. One day I'll take the diary home. And this goes till like 2, 3 or 4 days. And then it's gone, right? So, but the very first day we will write the name of Harsh and let's suppose Shriyasi, right? We write the names there. So, that diary belongs to both of us. So, antigen will be what? Antigen will be A and B. So, blood group will be A and B. But sir, now antibody what? I'm friends with both of them. Can I hate anyone? No. So, there will be no antibody. See, either I can hate A or either I can hate B. But both of them are now my friends. Both of them are now my friends. So, I can't hate anyone. Clear to everyone? Great. Exactly. Now, this has no name. Just I used to do like with my copies when I don't used to do the homework. So, no name. That means he can hate everyone. Anti and anti-B. The blood group will be O. The blood group will be O. And you write here, this is the case of codominance. So that whenever you revise, you don't forget. This is the case of codominance. Clear to everyone? Did you loved my session? Okay, let me revise one thing, what things we have discussed. So, today we have started the Post Mendelian Genetics. In Post Mendelian Genetics, we did the intra-gene interactions or interallelic interactions. In the first thing, we discussed about the incomplete dominance. We had few examples like Snapdragon, Mirabilis, Jarlapa, and the color of fowl. Okay. Then we came here, which was the case of codominance. In codominance, we discussed what? We discussed the sickle cell anemia, we discussed the human blood thing, and we discussed this black and white cow, black bull and cow. Then we came to the multiple allelism. In multiple allelism, we discussed the human blood group system. Okay. Now, in the next class, we will start with the RH factor. We will do the RH factor, we will do the blood transfusion, and then we will start with the pleiotropy. Okay. Clear to everyone? So now what you have to do is that I'll update the notes in the sheet. I'll put the link in the comment section just after the class. Download these notes from your end and try to write it, because when you're going to write by yourself, the concepts will be more clear, and revise. Take a rest for 15 to 20 minutes then, and let's gadgets is going to work very fine. Okay, till then, bye bye everyone, see you. DPP Vinika, I am making the DPP. Okay, so it will take little more time, but definitely I promise I am gonna give you. Okay, thoda sa wait kar lo, just wait a little bit more. Okay, 2 to 3 days I am making it. I am searching the questions basically. I don't want ki kahin se bhi utha ke I'll just put up the questions. I want basically, how do I make questions na? I first of all solve them. If I'm having, I put the timer of 30 to 40 seconds. If I'm taking those questions to solve like more than 30 seconds, that means that questions you are going to be stuck. So I try to give those type of questions in which you can get stuck. Okay. So it is creating little delay from my side. So sorry for that, but I'll provide you the DPP also. Okay, everyone? Great. Chalo. Bye-bye, everyone. See you in the next class.